So today it's the beginning of the third week of the program, and we will continue talking about genomic variation. But before we begin, let's see again our progress. This is the progress from the beginning of the program. You can find it in this URL. And I can see that on August 2nd and 3rd, there has been some progress in the educational side as well. It's the yellow one, and the gray one is the progress made on the server side. Today it's August 6th, and here are the participants that have made the most progress. You can see it for yourself as well in our server side. So before we begin, let's remember what we covered in the previous lesson. The previous lecture was about germline and somatic variation. Germline variants are the variants that we are born with. They are found in every cell of our body, and they are present in eggs and sperms, and they are inherited from parents to their children. While somatic variants are variants that are acquired during our lifetime in non-germline tissue. They are not transmitted to offspring, but instead they are passed on only to the descendants of that mutant cell. In the previous lecture, we focused in point mutations, and these are mutations that affect only a limited number of nucleotides, and they often refer to a change in only a single nucleotide. So we have synonymous or else silent mutations which don't change the amino acid sequence, like this one. We had the valine, and now we have, after the mutation, we have a valine as well. We have missense mutations, which results in an amino acid substitution. Like in this example, we had the proline and we have a threonine. We have the nonsense mutations, which are point mutations that generate a stop codon. So we have a prematurely terminated protein. And last, we have the frame shift mutations, which are small insertions or deletions that change the open reading frame of the protein. So we have a whole new protein product. Then we talked about the challenges of sequencing tumor DNA because the tumor genome is completely fragmented. Each cancer cell has its own genome, and sometimes we sequence the DNA from adjacent normal cells. So the usual practice when we want to detect somatic variants is to compare tumor to its paired normal DNA. And finally, we built together a pipeline in our server side using a tumor normal pair. These are the data sets, and we started the generated variation files together. Here is the URL to the pipeline that we built last time. So, moving on to today's lecture, we will talk about another type of variation, not point variation. It's the non-point variations. It's the copy number variations and the genomic rearrangements. And these are often more than 1,000 base pairs long. The umbrella terms that describe this type of variations is structural variation or genomic rearrangement. And as you can see on the figure, here we have duplication, which is when a segment of a chromosome is duplicated, as its name says. We have inversion, which is when a segment of a chromosome is reversed end-to-end. We have deletion, which is, again, easy, when a segment is deleted. We have insertion. Insertion is when a segment of a chromosome is removed and is inserted in another chromosome. And we have translocation, which is when two chromosomes exchange segments. I believe that the most well-known translocation is the so-called Philadelphia chromosome, and this is a genetic rearrangement in chromosome 22 of leukemia cancer cells, and in particular in chronic myeloid leukemia cells. So this chromosome is defective. This is the Philadelphia chromosome. It is defective and it is usually short because of translocation of genetic material between chromosome 9 and chromosome 22. So we have this translocation, and this segment of chromosome 22 goes here in chromosome 9, where this segment of chromosome 9 goes here in chromosome 22. This is the Philadelphia chromosome. It doesn't do anything, and it contains a fusion gene. This is a fusion gene. It's when two genes are together after such translocation, and it's called BCR-ABL1, and yeah, it's generated from this translocation. So, of particular interest is a subgroup of structural variations that we talked about. It's the copy number variation, and it is a phenomenon in which sections of the genome are repeated, and the number of repeats in the genome varies between individuals in populations. Specifically, it is a type of duplication or deletion event that affects a considerable number of base pairs, usually more than 1,000 base pairs. So here we can see that this is the normal, but here we don't have a C, we only have one copy of C, a deletion, and here we have a duplication. In this chromosome, we have two C segments, so we have 3 copies of C. This is a copy number variation. An interesting example of copy number variations is variation in the AMY1 gene. AMY1 protein that digests complex sugars. So, in every person, AMY1 gene's number of copies can vary from 1 to 20 depending on the person. And there is a study that suggests that having a low copy number of the gene favors obesity. So if we have only one AMY1 gene, we can't digest complex sugar that well, so we are more obese than a person that has, for example, 20 numbers of this gene. Okay, so the classic methods of mutation detection, such as Sanger sequencing, are usually unable to identify large genomic rearrangements, so we use other methods that can sometimes they can detect single nucleotides variation as well, but we can't detect the copy number variation with Sanger sequencing. These are some standard methods that are used for structural variation detection, but all these methods they are limited because they are designed to target either one or a few specific loci. We can't use them in order to detect structural variations throughout the whole genome. So for this course, we will study more high-throughput methods, and we will start from the SNP array. So before I continue, do you have any questions? We just talked about variations, and I think that before we go on to more details, you can make any questions. You can write on the chat or unmute yourself. Okay, I take it as a no, so let's go on. Okay. A SNP array is usually used for the detection of more common alleles of single nucleotide polymorphisms that derive from genome-wide association studies. Okay, someone says that there is a lag in the voice. I don't know what I should do. I am so sorry. Okay, I will go on, and hopefully you can hear me, everyone. Okay, so we usually use SNP arrays for the detection of common alleles, of common nucleotide polymorphisms that contribute to the development of a phenotype rather than alleles, rare alleles that predispose to phenotypes. And SNP arrays are extensively used in genome-wide association studies. Okay. But they can also be used to detect copy number variations by generating a virtual karyotype using software in order to determine the copy number of each SNP. So here in this diagram, the dots are SNPs that have been found using the SNP array. And we have compared them to a normal genome. So here, as you can see, the mean here for everyone is the green line. Okay, do you want me to pause a little and continue in 2 minutes? Maybe someone can leave and rejoin. Okay, I will pause for now, and I will continue at 11:15. Okay, I hope the problem is resolved for everyone. And in any case, we'll make a pause before continuing to the next slide, to each next slide. But I think we should continue. So we were talking about SNP arrays, which are a type of DNA microarrays that are used for the detection of common single nucleotide polymorphisms. And we can use them by reading the frequency by which we find the single nucleotide polymorphism. And we can use them in order to detect CNVs, copy number variations. So we have to generate a virtual karyotype using the software in order to determine the copy number of each SNP on the array. And then we have to align the SNPs in a chromosomal order. Here we have the chromosomes from 1 to X, and each dot represents a SNP. And so these SNPs are compared to a normal genome, and we have the logarithmic of their division. And here, the green line, it's the median. As you can see everywhere, the median is the same. But in chromosome 21, we have the median, which is the red line, which indicates that we have one more copy of chromosome 21. This is the Down syndrome or else trisomy 21. And in this slide, we have another example. Can someone, okay, most of you are biologists, I guess, but can someone tell me what this could be? Yeah, this is a male. Yes, great. So yeah, this is, again, we have the median, which is normal, but here we don't have two X's, we have an X and a Y, so we have a smaller median. So with the development of next generation sequencing, we have found more ways to detect structural variation. For example, we can detect it in a similar manner as with SNP arrays, but instead of comparing the SNPs, we can compare the coverage in the disease and the normal samples. So in this example, we have a homozygous deletion, a hemizygous deletion. This is the normal here, and here we have gain. So for this method, it is assumed a random distribution of reads from sequencing, and the divergence from this distribution is investigated in order to discover duplications and deletions. And as we said, regions with duplication will show higher read depth, while those with the deletion will result in a lower read depth. And up until now, we have only talked about copy number variation detection, but for the detection of other structural variations, we can use paired-end sequencing. And we have already discussed paired-end sequencing, but let's refresh our memory. Here we have single-end sequencing. This is when only one end of the molecule is sequenced. So the fragmented DNA is 300 base pairs long, and we only sequence 100 base pairs. We also have paired-end sequencing, where both ends of the cDNA molecule are sequenced. We first sequence this end, and then we sequence this end. So this technique allows the adaptable allocation of the segment, of the DNA segment during the task of mapping reads. by employing the distance between the two short sequence pairs. And this distance can help us identify other structural variations as well. So, apart from the read depth method, in order to identify structural variations, we have other methods as well. Read pairs examine the length and orientation of paired end reads. So, when we have read here, there is no structural variation, but when we have read pairs further apart than expected, we have a deletion. This is deleted. Here, with this way, if this read is here, while this read is here, we have a tandem duplication. And we have other methods as well. There is the split read method. They enable us to detect insertions and deletions, and the presence of structural variation is identified from discontinuous alignment to the reference genome. So a gap in the read marks a deletion, and a gap in the reference marks an insertion. And we have the de novo sequence assembly, and it can be applied with reads that are very accurate. However, the use of this method in practice is limited due to the insufficient length of sequence reads. We should have reads that are longer. You can find in the material files that I uploaded in the forum, you can find this publication as well as this one, if you want to study more about structural variation. So the most accurate approach in order to detect structural variation is using long read sequencing, because with this approach, reads are often tenths of thousands long, so the detection of structural variation is pretty straightforward. Right now, as we have mentioned before, long read sequencing is not that accurate for point mutation detection. So currently it is not used in a clinical setting, but with next generation technology advancement, we will be able to use long read sequencing in diagnosis too. And for now, the theoretical part ends, and we will go to the server side in order to do a demo pipeline for CNV detection. Okay, give me a moment, please. Okay, here is our server side. I'm sorry, let me minimize it. Here is our server side. CNV pipelines need a little more time to run than other pipelines. So I am so sorry, we will have to run the demo pipeline for this, for today's lecture. We will begin with pressing the start. Here you can examine the input files and the output files. I will tell you that these are files. I will tell you that these are files from tumor DNA sequencing, and these are the files that we used in the previous lecture. At first, we will perform PCR cleaning in order to remove duplicates, duplicate reads that have been inserted from PCR. Then we will align to a reference genome. And then we will use two CNV detection algorithms. One is the BRIC. You can see here what the algorithm does. And the other is CNVinator. Again, you can see here what this algorithm does, as well as the input and the output files. And then we will combine the results and press OK. Okay. I will refresh the page because sometimes this happens. And I will do it a little more quickly. Great. Here you can see info about what you should expect from the output. So here you can find from the demo pipeline, you can download all these files in order to examine them. Together we will see a little the 1000G VCF, which has, have opened it in Excel in order to be able to see it better. This is a VCF file like the ones that we saw together last time for the single nucleotide variations. This is from the 1000 genomes. So it's germline, it's not tumor. And we have 4 copy number variations, no, structural variations. We have chromosome position, its ID if there is one, the reference, what we should expect to sequence, and the alternative. So here, CN0 means we have a copy number of 0 copies. So we have a deletion, and inf means inversion. The quality, here this has been generated from Dale, which is an algorithm for structural variation detection, which doesn't generate any quality. And we have some information which you can see in this section of the header. You can see what each section says. So SV type, it's here, it's the type of structural variant, which as we said is deletion, inversion, and deletion. CIN, if you see here, is the confidence interval around end for imprecise variants. So structural variations are many times imprecise. So we say we have a deletion of exon of a gene, but we are not sure from where it begins and where it ends. We don't know the exact position of the beginning and the end. So we have a confidence interval, and we have the end, for example. So it says that it's in this position, it ends here, and the confidence interval is 0. So we know for sure that this variation ends here. Here in the inversion, we have a confidence interval of minus 130 and 130. So we have the end, which is here, but it could be 130 bases before or after, etc. And here we have other information because this is a file from 1000 Genome Project. For example, the allele frequency in some populations. For example, EAS is East Asians, EUR it's Europeans, etc. And feel free to explore the file by yourself. You can look in the header in order to see what each section means. And if you have any questions, you can as well Ask me whenever you want. Okay, do you still have a problem with no, this is not only this. I was just asking if you have a problem with lag, so we will move on to visualization. But please tell me if you have a problem with lag. One answers, I guess it's okay. Okay, so next we have this file, which is this one. We have two files from TCGA, from normal, and from tumor, from breast cancer, and they are in a seg file format. If we open this, I think I have the tumor file. Yeah. If we open the tumor file, which, as you can see, it's .seg, the seg file format is a tab-delimited text file that lists loci and associated numeric values. The purpose is to register the copy number value of its listed loci. So the first one is some ID or the ID of the sample, etc. And we have the chromosome, the beginning of the section, and the end of the region for which we calculate copy number. And here we have data from TCGA, which are usually data from array CGA, which is a microarray assay as well. And for the last two columns, we have number of probes and segment mean. And let me explain you what this means. Each probe registers an intensity. They are fluorescent probes. And this intensity implies the copy ratio estimate for that specific probe, which is relative to a baseline. It is usually represented in a logarithmic scale. So the number of probes doesn't mean a number of copy number. Someone is asking if this set file is generated by Free, the seg. As far as I remember, no, Freak gives CNV files. And okay, let me look at it. Yeah, it is in a yes. Answers, thank you. Yeah, it is a visualization output as ASS, and it's a similar format. Okay, so again, let's go to the number of probes. This does not mean the copy number. It just tells how many probes were in this particular region, and each probe has its own intensity, which gives a measure of copy number, a copy ratio. So the segment mean is segmentation groups together adjacent probes likely to have the same copy number. So in this format, the segment mean is the arithmetic mean of those probes' logarithmic copy ratio values. So this indicates the copy number. Here that we have a negative, no, 0 means that there is no copy number. negative means that there might be a deviation, and positive means that there might be some gain in this particular region. So here that this is a relatively small number, maybe there is no deletion, but we can't be sure by this. But here, which is above 1, it's for sure, it's certain that we have a deletion. And I will show you these files in the IGV. IGV is a genome browser that we use in order to visualize such files. Let me share with you the URL so that you can be able to download the IGV. You can download it either on a computer, a Windows computer. Here I run it on Linux, and it runs on Apple computers as well. Okay. So when we run IGV, we have here we can select which reference genome we want to study. Here we have AG38, and from here you can download other genomes as well. And here are the reference genes, and here is the reference genome. I have already loaded these two files, and by rename track, I have entered a new name because we have another name. And you can load from here, file, load from file. So, when we visualize seq data, we have either white, blue, or red. The lighter the color, the more close to 0 it is. So when we have white, it means we don't have any gain or deletion. And when we have blue, it means that we have a deletion. And when we have red, it means that we have some gain. Here that we have a very dark red, it means for sure that we have some gain. As you can see, the normal file doesn't have almost no deletions or gains at all. I forgot to say that gray is areas for which we don't have any. Okay, someone asked what are the fluorescent probes? These are okay, probes are small molecules that target a specific region, which we use them in order to detect variation, either single nucleotide or copy number variation. And fluorescent probes, I don't know exactly what the chemistry is, but we put some fluorescent substance which gives a light. So the brighter the light, it means that the brighter the light, the greater, depending on our experiment, if we look for CNVs and we have a bright light, it means that we don't have a CNV. Because, no, excuse me, that's a mistake. If we have a deletion, we won't have a light at all. If we have, again, we will have a bright light. And this will, we have to compare it to a normal in order to be able to say that this is an intense light or not. Someone else is asking which file I loaded. Yes, it's an output of the demo CNV. Yes.
So, Despina, maybe I can just add to explain what these demo pipelines are.
Yes, thank you, Leah, very much.
Okay, so basically the purpose of the demo pipeline is to illustrate what the expected output would be so that we understand that by loading a tumor and a normal sample, we can have an output that gives us either an insertion or a deletion of a region. So we know the length of the region, we know the significance of that region, and we know whether that's an insertion and a deletion. And as we can see in this example, we have a comparison against the reference genome, and some areas are not present even in the normal sample. But then in the tumor sample, we have a lot more aberrations or variation in terms of copy number. And so the interest in our discussion today is to understand the scores and understand what is in the file, and then visually see how that aligns to chromosomes, to genes. What would be the interpretation of this ultimately in the biological meaning of this? And then what we want to do in the some of our future lectures is discuss more specifically why, for example, in this pipeline you see two algorithms that are supposed to do the exact same thing. They're supposed to identify these copy number variation positions. And then why do we have this table to combine the outputs of multiple types of analyses? The reason is that, as you will read in a lot of papers referencing CNVs, you have each algorithm essentially produces a whole range of differences. To reconcile these differences, we would want to take the overlapping results. So that is just a general explanation of why we are using the demo for now. And as we go further, we'll talk about the differences in the different algorithms, unless you want to mention today something, Despina, about the difference between the algorithms. But basically, the idea is to just show this is the conceptual output, this is what you would be getting at the end, and this is what that might be interpreted as. And then we'll go backwards and we'll talk a little bit more of the technical inputs and outputs of those specific algorithms.
So the
Yes, thank you, Leah.
sorry, I have a quick question here. So
Yes, yes.
as I understood, so the point is that if we use more than one algorithm for analysis, so the outcome will be a kind of quote unquote average of those two algorithms taken together, or will the output be three outputs, one for algorithm one, second for algorithm two, and third for a kind of combined outcome? So how would it be?
Yeah, so as you will see, for example, as Despina was showing in the VCF file originally, it tells you which algorithm produced that copy number variation. So several things are going to be important here. Which algorithm, based on which genome, and also based on sequencing method a lot as well. So you would have multiple factors affecting your output. And therefore, the safe way to do this is to look at multiple algorithms that might have some overlap. I don't know if, Despina, you have any kind of a statistic on how much the results of these algorithms would overlap, but we can probably talk about that in the next lecture. So the safest would be the ones that are identified common between multiple algorithms. Now, about other ones, that's where your understanding of the method would be critical. So if you know about how the method works, in a specific situation, that specific algorithm might be your best pick, but maybe not all the time, because copy number variation could be studied in the context of many different conditions, many different genomes. Cancer is just one of those examples. And so, understanding which method to use, which method of sequencing to use, what reference files to rely on, etc., is going to guide your selection process. I mean, in this case, we're looking at tumor and normal sample in TCGA, which was whole genome sequencing. So this is kind of a unique case in terms of the variety of applications of CNVs.
Thank you, Leah. Do you want to see the CNV in a little more detail? We can go here. We can choose chromosome 8, which you can see has a band of very dark red here. And as you can see, if you hover over the band, you will see that it has a value over 1, which is it's not very close to 0 in a logarithmic scale. So here we have a gain in the last section of the chromosome 8. And here we might have a loss in the chromosome. It's minus 0.5. But it would be, okay, here we have minus 1.2. So for this segment, we are more certain that there is a loss than for this segment. But as I said, if we combine many algorithms together, we can see which sections overlap in the algorithms. Unfortunately, I don't have a statistic to tell you what is the overlap between these algorithms, but I can search for it. Okay, I see that Leah answers the questions in the chat. Thank you very much. So I don't have anything else to ask you, to show you here in the IGB. I think that this is the time for questions in today's lecture, either in the IGB or the lecture, the theoretical part. Please feel free to ask me either in chat or by unmuting yourself.
Is this a male sample?
A male sample.
Male,
Okay.
right. Sample
Yeah.
comes from male because it seems that there's a deletion of Y chromosome.
Yes. No, it's a deletion of Y chromosome. So
Sorry?
it's a deletion of Y chromosome. In the previous slide that we had a male, it was only the X chromosome. We didn't have the X and the Y together. Here we have a deletion of the Y chromosome, so it's a female. And it's breast cancer, which is most common in women. So yes,
Got it.
we have a female.
And Despina, could you maybe show any specific genes or regions
Yes.
that might be relevant to breast cancer in this example?
Yes, so before we begin, so in chromosome 17, we have the most genes that are relevant to breast cancer, to cancer in general. So I went to TP53 that we have talked about already, but you know, it's very close to 0, so maybe we don't have a loss or a gain, but let's see BRCA1 as well. BRCA1 the same. Unfortunately, we don't have what we call loss of heterozygosity in the tumor in a significant gene relevant, unfortunately. Yeah, maybe it's good for the woman that has the cancer, but you know, in order to show the example, we don't have. But if we had the loss of the tumor allele, the BRCA allele, here we would have a dark blue color, which would indicate that BRCA1 is completely lost. Okay, someone is asking if it is possible to do CNV analysis with whole exome sequencing. These are two different techniques. The fluorescent probes are used in DNA microarrays, in SNP arrays that want to target a specific region. But if we have array, we can target some regions together. So this is a high throughput experiment as well. We can perform with sequencing. Whole genome sequencing would be the best, but we can perform CNV analysis with whole exome sequencing as well by using the approaches that we discussed during the lecture. These approaches, we can use the coverage in order to perform copy number variation analysis. We can use the distance between the read pairs. If they are closer than they should be, we have a deviation, and if they are far apart than they should be, we have an insertion. So maybe I didn't explain it very well. This is the sample, and this is the reference genome. In every picture here, we have the samples and the reference genome, and we can use the split read method, which here it is a read, but as you can see, our read has when we map it to the reference genome, the half is found here and the other half is found here, which indicates that this whole area has been deleted. And we have the novel sequence, the novel assembly of the sequence. So here that we have a novel sequence insertion in the reference genome, these reads can be assembled in order to make this to de novo make this sequence, and we can say that this new sequence has been inserted into the reference. It hasn't been inserted. It has been inserted into our sample. So these are the four methods that we use in order to detect copy number variation using whole NGS data. Do you have any other questions?
I have one question, and maybe a little bit more like a discussion, more for a discussion. So I was wondering, like, once we get more like hands-on experience of using this pipeline, so if we could, ourselves do some little analysis where we could take like different coverages of the sample files, and using maybe a couple of different algorithms which are available in the pipeline, we can come up with some kind of outcome or analysis where we could show that how by decreasing the coverages, how we can impact how effective or ineffective the algorithm becomes as far as detection of CNVs is concerned. Because that way we'll be able to kind of get a better understanding of what is the good coverage to detect CNVs and how many CNVs actually are kind of visible once we follow a couple of algorithms. So again, just a little bit discussion over here.
Okay, so when we use coverage, it's not that straightforward. So we don't say that in this region of the reference of the genome we have more coverage than in the other region, because each region is not it doesn't have the same it has the same difficulty to sequence. So, for example, it's in some regions that we have a great GC content, we will have a lower coverage. So we have to perform some normalization techniques in order to be able to say to detect CNP based on the coverage. So it's not that straightforward. We won't be able to load the BAM file in the IGV and say that, okay, here I don't have so much coverage, so I have a deletion. I know that maybe that's not what you were hoping.
Well, maybe I can just jump in here and say that a couple of links that I pasted in the chat are publications where people did that, an attempt to compare how these different algorithms perform, and then also do that in general, but also do that in the context of a specific challenge, like, for example, in cancer. So I think that your idea is definitely relevant. People have been benchmarking these different algorithms, but you have to just maybe more keep in mind what type of sequencing, so not just coverage in general, but for example, specific challenges exist for whole exome sequencing, where you're only sequencing the exons, but the coverage within the exon might be of different levels. And then how do you maybe compare this to whole genome sequencing, which in overall will have lower coverage than whole exome sequencing. And then I think that also in terms of what type of comparison are we trying to look at? Are we trying to look at false discovery rate? Are we trying to look at specificity or even more detailed comparisons? For example, because of differences in coverage, many of the algorithms struggle to combine smaller portions into a full copy number. Because when we say copy number variation, typically, links are important. So, you can detect shorter sections easier than longer sections. And that's where a lot more statistics comes in, and an understanding of, what would be the distribution, what is the expected coverage, things like that. So I think in general, the idea might be appropriate, but before jumping into actually doing something like that, I would suggest reading several of these articles. These are just 2 example articles, but there's obviously a lot more out there that, maybe we can learn from what other people did and what other kind of considerations are there when this type of benchmarking is performed for CNP.
Thank you, Elia. Is there any other question that you would like to ask?
So is copy number variant, understanding only relevant to cancer, or are there good examples in diseases or conditions other than cancer?
Mm-hmm. Okay, so
I know it's a biology question, but
yeah, as I showed you before, there is the Down syndrome, which is again in the whole chromosome 21. So we have 3 copies of chromosome 21. This is the copy number variation as well. Copy number in human genetics, it's detectable in every genetic disease. I show you another example. Okay, let me go in this slide. This is an example of copy number variation as well. This is the AEMY1 gene, which digests complex sugars. And the this is a copy number polymorphism, we could say, because each person has 1 to 20 copies of this gene. So it has been suggested that a person that only has 1 copy of this gene cannot digest properly complex sugars. So it has a tendency to obesity, while another person that has more copies has a greater, is not obese. So copy number variation is in every genetic disease, but in cancer, in tumor DNA, it is more detectable because tumor DNA is completely fragmented. And we might have 5 or 6 or 0 chromosomes in a pair. For example, we might have 6 chromosomes, 21. So this is why we look at it in the tumor DNA for our example today. Does anyone have another question? Thanks everyone, but I can wait a little if you have another question. Aya has left the meeting, but I would like to thank him as well for all the support. Okay, thank you, Dr. T. And if you have any question, you can add me on LinkedIn, and you can send me any question you have. I don't have any problem to answer them, but maybe I won't answer straight away. You will have to wait a couple of days. I have shared some articles with you in the forum section. Before every lecture, I upload, I share a link, a Dropbox link with you, which have the slides of the lecture and some other material. And today, there are 3 articles as well. Yeah, and as Pratim says, you can also post questions in the forums, and everyone from Biotech can answer, not only me. Okay, yes, I can go to the forum. Okay, this is our educational site. This is my profile. I am a member of OmicsLogic Genomics. And this is the forum here. And you can click on your progress as well, as I showed you. And we have the recordings posted by Sahil. Thank you, Sahil. And here in this topic, it's the material for every lecture. So tomorrow, I will post again for tomorrow's lecture. And you can download everything from here. So if you don't have any other question, maybe we could say that we call it a day for today.
I have a quick short question, if you
Yeah,
don't mind.
okay. Thank you.
No problem. Thanks a lot. So, I think I asked the question earlier, but at that time it was more pertinent to SNPs. And my question was that when we detect a SNP, especially in conditions like cancer, let's say in a testing lab, then we confirm it using Sanger technique. So here, what is a workflow? Like if you find a CNV, let's say copy number variant in our panel by NGS, then how do we reconfirm this finding, like by some other
Yes,
orthogonal?
yeah, thank you for your question.
Okay.
There are other techniques as well. For example, here these are the standard methods for structural variation detection. In our lab, for example, okay, we have what we call hotspots. There are some 2 CNVs in BRCA1 that are very common in Greek population. So you can understand we want to perform next generation sequencing, which is very costly in order to detect these specific structural variations. What we do is we perform, where is it? Yeah, multiplex, quantitative multiplex PCR. I don't know exactly how this works because I am not a biologist, but you only target a specific region that you want to target. So if you find something in NGS, you can confirm it then by another method, one of these methods, where you target only the specific regions that you want.
Okay. Okay. Thanks. Thanks for the answer.
Thank you very much. So if you don't have anything else, I will see you tomorrow. Thank you very much for today.